Hi, this is Dong Lim, and I'll walk through the topic of this scientific poster. Uh, I did an experiment to understand the role of extracellular signal-regulated kinases, which is called ERK, within the nucleus accumbens for amphetamine-induced sensitization. Amphetamine is a psychomotor stimulant that mimics the sympathetic nervous system. In other words, it induces some behavioral activation. The interesting part about a psychomotor stimulant like amphetamine is that it induces sensitization. What sensitization means is that the repeated administration of a drug increases some of the drug effects. It was studied in numerous past papers. Of all, what seems to be most relevant with the sensitization was related to the ERK, extracellular signal regulated kinases. ERK interacts with some phosphate groups and alters transcription factors. A recent study from Fuchs demonstrated that ERK activation might be essential in understanding sensitization because the p-ERK activity showed contradicting responses for acute and repeated administration of stimulant drugs. Our targeted brain region was nucleus accumbens, which is located in the ventral striatum. The striatum is an essential brain part which acts as a motor interface that connects different brain parts, including non-motor related brain regions. For our experiment, we observed three types of behavioral changes: locomotion, head movements, and stereotypy. We hypothesized that activation of ERK within the NAC would be essential to the expression of amphetamine-induced behavioral sensitization, and more precisely, an amphetamine antagonist U0126 would prevent such sensitization. For this lab, we used two male adult rats. Rat 1 was the experimental and the Rat 2 was the control. To inject amphetamine directly into our designated area, we drilled two holes on the rat's brain and placed a cannula, which can transfer the drug. To do that, we carried out a craniotomy. Their behavior changes in the cage were observed and recorded for a 2-hour session. For locomotion, the total time span for horizontal movements, which uses all four legs movement, was recorded by our group members. For both stereotypy and head movements, we used a scoring table that was given from the PSY111 course, with a total of 4 lab sessions, excluding the surgical procedure and the recovery time. The first lab was the injection of saline, the second lab was the injection of an acute amphetamine, and the third lab was the sensitization part where the repeated administration of amphetamine injection was done. And the last lab section was about microinjection of U0126 to see any behavioral alternation that was present as a sensitization. To make sure that our microinjection was placed correctly, we carried out a histological procedure using a vibrating blade. The brain sections were stained by crescent violet that shows the cell bodies. Statistical analysis was done using SPSS software, and we ran some t-tests and ANOVAs. The statistical analysis showed that repeated administration of amphetamine had effects for all three behaviors, but only head movements and stereotypy seemed to develop sensitization. Then, using the software, the alternation in those behaviors were analyzed, and it turned out that the only head movements were altered by microinjection of amphetamine antagonist U0126. Since our histological results showed the right location for the microinjectors, we could assure that the antagonists induced such changes. Therefore, it could be said that the activation of ERK within the NAC is essential for head movements, the behavioral sensitization by amphetamine. However, there were some limitations as well. The post hoc analysis demonstrated that there were some significant effects for a few time sessions within the first hour. This might be due to the lower potency of U0126 because certainly other behavior sensitization was altered in the beginning. Therefore, it might be ideal to have another study with a higher efficacy antagonist for ERK. Although some behavioral sensitization was altered by ERK inhibitions by U0126, it is also difficult to conclude any causation because of the reaction that happened in the rat's brain was not observed at a cellular level. Therefore, the neuromechanism is still unclear. Furthermore, the connectivity to the striatum is not limited to the dopaminergic pathway where ERK acts on. So in the future, investigation on other parts of the striatum might give a better insight into the ERK role. More generally, studying other types of neurotransmission signals would help to better understand the amphetamine-induced sensitizations in terms of behavior. This experiment was effective because inhibition of ERK certainly altered the sensitization of amphetamine. I want to say thank you to our instructor Leo and also Dr. Zalumsky for conducting the experiments. Thank you.